This is our presentation on caloric restriction and aging, insights from the NIA Rhesus monkey study. Our article is called The Impact of Caloric Restriction on Health and Survival in Rhesus Monkey Studies from the NIA study that was published in 2012. So here's a little bit of an introduction on our article. So the authors wanted to study the impact of caloric restriction in rhesus monkeys to determine if there was a significant effect of caloric restriction on the onset of age-associated diseases and mortality. Past studies found that there had been contrasting results with the association of caloric restriction and mortality. Therefore, this research aims to find out if there is a separation between health effects, morbidity, and mortality, which has been shown in rodents in the past. So the research objective of this article is to see if the diet composition strongly affects the lifelong effects of caloric restriction in non-human primates. So next we're going to move on to the background and objective of this article. So the objective, as stated previously, this article aims to research if caloric restriction, a reduction of intake of nutritious diet, extends lifespan in rhesus monkeys. So this article reports that caloric restriction has been reported to prevent the occurrence of many chronic diseases, alongside with beneficial effects on immune system, motor coordination, and resistance to sarcopenia. However, there were contrasting results at the National Institute of Aging that reported that CR has not improved survival outcomes, but this differed from what was going on at the ongoing study at the Wisconsin National Primate Research Center, which reported that they had improved survival associated with CR in adult rhesus monkeys. Furthermore, in this study, CR was initiated in monkeys in varying ages to test the impact of age of onset of caloric restriction on its biological effect. So this includes young onset and old onset monkeys. In both studies, they used longitudinal measures to measure metabolic health and overall function. This includes measuring levels of triglycerides, cholesterol, and glucose with age for both genders. This research article studied survival curve for old onset monkeys and survival curves for young onset monkeys in order to generalize if CR has an impact on mortality and age-related deaths.
And I was in charge of the methods part of our presentation, so I'll be talking about details about the subjects, the research design, and specific procedures followed during the study. This 23-year-long study was done on rhesus monkeys. I believe because their genome is a 93% match approximately for sequence identity compared to the human genome. They also have common anatomical aspects with human and common effects from aging and development. I included a monkey census chart from the original report where I detail the old onset and young onset groups of male and female monkeys and detail the subdivisions within those two bigger groups. I also talk about the medical procedure policy, one of which being that the NIA initially did not treat cases of long-term chronic disease in monkeys. However, in 2004, the policy was modified to allow treatment and prevention of endometriosis after it ran a bit rampant in the female monkey population. This was done to reduce the possible bias of the disease on the results. I also included details about the monkeys' diets, which were that they were fed a diet consisting of 56.9% carbohydrates, 17.3% proteins, 6.5 to 9% fiber, and 5% fat. I also included details about the sample collection and storage. Blood samples from the monkeys were obtained under ketamine and anesthesia after the monkeys fasted overnight. Before analysis, serum samples were stored frozen at 80 degrees Celsius.
Moving on, we will be discussing the results and conclusions of the study. As a reminder, this study focuses on understanding how caloric restriction, or CR, impacts lifespan and health outcomes in Rhesus monkeys, a long-lived primate species, by investigating both young and old onset CR groups. The study explores whether CR provides consistent health and longevity benefits, as seen in short-lived animals like rodents. Let's begin. Old onset caloric restriction, a lifespan analysis. So this first section focuses on the effects of caloric restriction on the lifespan of monkeys that started CR at an older age. Between 16 and 23 years old, the goal was to determine if CR could extend lifespan when introduced later in life. Monkeys in the old onset group were divided into two groups: a CR group that consumed 30% fewer calories, and a control group that received portion-controlled diets to avoid obesity, ensuring that both groups maintain nutritional adequacy. The Kaplan-Meier survival curve on this slide show the results. There was no significant difference in survival rates between the CR and control groups, as indicated by a value of 0.934. Both groups experienced similar age-related mortality patterns. Interestingly, males in both CR and control groups lived significantly longer than females, with a p-value of 0.003. This sex difference is consistent with previous research in Rhesus monkeys and could be due to hormonal or genetic factors. 4 CR monkeys and 1 control monkey lived beyond 40 years, exceeding the median lifespan of 27 years previously reported for captive monkeys. However, the authors attributed this to excellent husbandry conditions rather than the effects of CR. Post-mortem analysis revealed no significant differences in the cause of death between CR and control groups. Common age-related diseases such as cardiovascular disease, amyloidosis, and cancer were equally represented in both groups. This suggests while CR does not extend lifespan, the study's environment may have contributed to the overall improved longevity. In conclusion, CR did not extend mean or maximum lifespan in the old onset group. These findings contrast with earlier studies in rodents highlighting the complexity of translating CR effects to long-lived primates. Old onset caloric restriction, metabolic health. Although CR did not extend lifespan in the old onset group, it showed significant benefits for metabolic health. This part of the study evaluated markers such as triglycerides, cholesterol, and fasting glucose levels, which are important indicators of cardiovascular and metabolic health. This first graph, figure 1b, shows triglyceride levels over time. Triglycerides increase with age in both CR and control groups, but CR monkeys, particularly males, had significantly lower levels, a p-value of p=0.026. The second graph, figure 1c, highlights cholesterol levels. CR males also had lower cholesterol levels than control males, especially after age 30, with a p-value of 0.02. This finding suggests that CR slows the progression of age-related increases in cholesterol. Fasting glucose levels shown in the third graph, figure 1d, were numerically lower in CR monkeys. This reduction was statistically significant in males. It showed a p-value of 0.04, but not in females. Interestingly, 5 glucose measurements exceeding 100 milligrams per deciliter were excluded from the analysis to avoid skewing the results, reflecting rigorous data handling. Plasma isoprotein levels, a marker of oxidative stress, were significantly lower in CR males compared to controls. It had a p-value of 0.009. This reduction indicates that CR may mitigate cellular damage caused by oxidative stress, which is a known contributor to aging and chronic disease. These metabolic improvements suggest that while CR did not extend lifespan, it positively impacted health markers associated with aging. This underscores the potential for CR to improve quality of life, even if it doesn't increase longevity. Young onset caloric restriction, lifespan, and cancer incidence. Next, we'll look at the effects of CR in monkeys that started the diet at a younger age. The goal was to determine if early CR could extend lifespan or reduce the incidence of age-related diseases. Survival data for the young onset group revealed no significant differences between CR and control monkeys. Looking at figures 2a and 2b, approximately 24% of the control monkeys and 20% of the CR monkeys died of age-related causes, suggesting that CR had a minimal impact on overall survival. However, when we examine disease incidence, figure 3a, we see a striking difference in cancer rates. As shown in the bar chart, no cancers were observed in the CR group, whereas multiple control monkeys developed cancer, with a mean age of diagnosis of 22.8 years. This reduction in cancer incidence was statistically significant at a p-value of 0.028. This aligns with findings from rodent studies where CR has consistently reduced tumor growth. Interestingly, CR did not reduce incidence of other age-related diseases such as cardiovascular disease or diabetes. These findings suggest that while CR benefits are disease-specific, they do not translate to a broad reduction in all age-related conditions. In conclusion, CR significantly reduced cancer risk but did not extend lifespan or delay most other age-related diseases. This highlights the complex and selective nature of CR's health benefits. Young onset CR, metabolic and oxidative health markers. This slide focuses on the metabolic and oxidative health markers in the young onset group. Researchers tracked fasting glucose levels and oxidative stress indicators to evaluate the broader health effects of CR. Fasting glucose levels, shown in the graph, figure C2C, were not significantly different between CR and control groups, indicating limited metabolic benefits in this cohort. However, CR males exhibited marginally lower triglyceride levels with a p-value of 0.051, suggesting a potential trend toward improved lipid metabolism. In terms of oxidative stress, plasma isoprotein levels were lower in CR monkeys compared to controls, indicating reduced oxidative damage. This finding is consistent with old onset group and highlights a potential protective effect of CR against cellular aging. These results suggest that while young onset CR may not drastically alter metabolic markers like glucose, it does offer modest benefits in reducing oxidative stress. This supports the idea that CR can enhance some aspects of health, even if it doesn't extend lifespan. To summarize, this study provides important insights into the effects of caloric restriction on aging in rhesus monkeys. For the old onset group, CR did not extend lifespan. It did, however, improve metabolic markers, particularly in males with lower triglycerides, cholesterol, and oxidative stress, and no significant reduction in age-related diseases, similar causes of death in CR and control groups. For the young onset group, CR reduced cancer incidence with no cases observed in the CR group. However, it did not significantly extend lifespan or reduce other age-related diseases. Limited metabolic benefits were observed with lower oxidative stress, but minimal impact on glucose levels. Overall, these findings suggest that while CR improves certain health markers, its ability to extend lifespan in long-lived primates remains inconclusive.
So, in conclusion, according to the results of the experiment, calorie restriction monkeys did experience improvements in overall health and function. However, they did not show an increase in longevity in comparison to control monkeys. Their findings suggest that study design, cultivation, and diet structure are significant components in prolonging CR monkeys' lifespan.
Let's dive into the pros and cons of the ongoing study at the Wisconsin National Primate Research Center. First, the pros. One of the key findings is that the caloric restrictions has led to a 30% improved survival rate in adult monkeys, specifically those between the ages of 7 and 14. This is significant because it highlights a potential link between caloric restrictions and increased longevity. Contributing to our understanding of aging, while the results are promising, they also invite contrasting viewpoints. Some argue about how broadly these results can be applied or replicated, especially in other populations or species. And now let's talk about the cons. One major limitation is the uncertainty in drawing a definite conclusion about CR's impact on overall mortality rates, readers and researchers are often left wondering, will we ever have a clear answer? To illustrate this frustration, consider a simple question like, what color is a cloud? The answer is clear, white. There is no theory suggesting clouds are neon green. But with CR and mortality, the answers are less clear, which can be unsatisfying for those hoping for straightforward outcomes. Ultimately, the study opens the door for deeper research and ongoing debate, which is both its strength and its challenge. Thank you.